if you see something that you think might be helpful. And then our notes are over here. All right, there you go. So we are in week 3 of the final term here at the Excel Center. So we're 2 weeks down and 6 weeks left to go. For the most part, people did really, really well on the question yesterday about the primase and the importance of the RNA primers to get DNA replication started and off to a good start. So good job on answering that. So this week, we're going to be covering essentially one set of notes over the next couple days. Hey, Junior, good to see you. Thanks for being here. And so the topic that we're going over is big. It's a little complicated. It's not quite as complicated as the DNA replication, but I'd say there's more steps and some more details that we're going to get into. And so let's go ahead and get started with our kind of first half of the notes for today. So these notes are called From DNA to RNA to Proteins, because that is what we're going to be talking about today and Thursday. The ultimate goal of the DNA code is to create proteins, which are small molecules that do a whole bunch of different jobs in our cells. So proteins are responsible for helping to build and maintain our cells. They do a lot of the functions in our cells. So all of those enzymes that we were talking about on Thursday, the helicase, the primase, the polymerases, all of those are proteins. And so the instructions to make those proteins is inside of our DNA. And so today we're going to talk about how and why we have to take that DNA code and turn it into another related structure called RNA. And then on Thursday, we'll get into how we take that RNA that we copied and turn it into a protein that's going to go off and do some sort of important job inside the cell. So DNA, hopefully everybody is pretty familiar with at this point. We know about our bases, our A, G, C, and T that make up our big strand of DNA. And segments of those bases are called genes. So a gene is a specific sequence of bases. It could be a couple dozen bases long. It could be a couple hundred bases long. But genes are these segments of DNA. And these genes contain the code in order to make a specific protein. So proteins also sometimes referred to as polypeptides. That's kind of a fancy scientific way of putting it. But each gene codes for one protein. So we have, like I said before, DNA is huge. There's millions and millions of base pairs in DNA. And so DNA, especially in the case of humans, which is kind of the main reference that I'm going to be making, in humans, we have hundreds of different genes, hundreds of different DNA sequences that each have the code for hundreds of different proteins. So each gene makes one specific protein. And the instructions for making that protein are found in the bases of those genes. So the different combinations of A, C, T, G is what gives us our specific protein. And today and Thursday, we'll talk about exactly how that happens. So proteins do a whole lot of different jobs in the cell. They're used to help maintain the inside of a cell. They're used to help build new cells, especially muscle cells have a lot of protein in them because there are big protein-based structures that need constant maintenance and repair and renewal that's done by other proteins. So proteins are extremely important for our body and for our cells. And the proteins inside of our cells that do the different jobs of the cell come from the DNA and come from the genes in our DNA. So this is why DNA is so important, because it has these instructions for making the proteins. So a general structure of a protein is a bunch of smaller segments called amino acids that are linked together. So just like in DNA, we had our nucleotides, that phosphate sugar base, and we put millions of those together and it gave us a DNA. Proteins are not made up of nucleotides. They're made up of things called amino acids. And actually, now that I think about it, I want to highlight that a little bit more. We're going to go like this and go like this. There we go. So proteins are made up of amino acids. So there's 20 different amino acids that are out there in the world. And depending on what type of combination of these that we put together, we'll get a different protein. So just like a gene is made up of a couple hundred bases, a couple hundred nucleotides, a protein can be made out of a couple hundred amino acids. It all depends on which protein you're talking about. Some are small, some are big, but all of them are multiple amino acids that all get linked together. So luckily, you guys don't have to know too much about the specific amino acids themselves, like what their structure is or what their names are. The main thing to know about amino acids is they all have the same basic structure. So they all have this sort of central carbon. And then one side is called the amino group because it has this NHH structure, this nitrogen piece. And then this COO group over here is known as the carboxylic acid group. So every single amino acid has these exact same three pieces. This carbon that's attached to a hydrogen, and this nitrogen group, and this carbon-oxygen group over here. Every single amino acid has those. Where they differ is this thing down here called the side chain. Each amino acid has a different side chain. The side chain could be just another 2 or 3 carbons linked together. It could be a ring of usually carbons and nitrogens together. It could be some carbons and some sulfurs mixed together. It could be a big branching structure of carbons. All sorts of different things can make up this side chain. And that's what, so there's 20 different side chains, and each one marks a different amino acid. But all amino acids have this same basic structure up here. Nitrogen group, carbon, oxygen, oxygen group, and this kind of central carbon and hydrogen right here. So luckily, you guys don't have to know all the different side chains and all the different structures because you guys are not Mr. Stanley, who once in college took a biochemistry class in which the teacher made us memorize every single structure of every single amino acid that is out there, all 20 of them. We basically had to know every one of these different structures down here. So unless you take a college level biochemistry course sometime in your future, you probably will never need to know that. But just know that amino acids have these shared structures, and they are the building blocks of proteins. So we have all these different amino acids with their different side chains. We can link them together in this bigger chain. And when they're first linked together, they're called polypeptides, because another name that some people give amino acids is peptides. So when we link them all together, we have a polypeptide, and then we fold that all up together. And when that gets all folded, it makes a protein. So a protein is a specific sequence and folding structure of all these different amino acids. But again, they're all amino acids, and so it's some combination of those 20. So in order to make these proteins that we're going to make, we need to take some amino acids, link them all together, fold them up, and turn them into a protein. So does anybody have any questions about proteins or amino acids so far?
No.
All right, awesome. So in all of this stuff that we're doing, this process of turning these genes into that protein, it all starts with our DNA, our double helix, double-stranded DNA. So DNA inside of a cell is found inside a structure called the nucleus. If you took Bio A, when we talked about all the different parts of the cell, you definitely remember us talking about the nucleus. It's this big structure in the middle of the cell, and its main job is to protect the DNA. So the DNA is inside this nucleus where it can be nice and safe. We don't want it floating around in the other cell with all the other stuff that's out there, because if the DNA becomes damaged or something happens to it, that could be a big, big problem for the cell down the line. So the cell wants to keep the DNA safe, so it stores it in this big structure called the nucleus. The one downside of this, however, is that proteins are not made in the nucleus. Proteins are made by other structures, other organelles, which are like little structures in the cell, called ribosomes. And again, if you're in biology A, you may remember that we talked about the ribosomes, which are these little protein factories. They can be studded across that weird structure, the endoplasmic reticulum, that we talked about a little bit in biology A, or they can be floating around the inside of the cell on their own. So ribosomes are all floating around in the cell. That's where our proteins are being made. So the ribosomes are like our little protein factories. But the ribosomes are not in the nucleus. Only the DNA and certain DNA-related proteins are in the nucleus. The ribosomes are on the outside. They're floating around in the rest of the cell in this kind of cell jelly called cytoplasm. So the instructions are inside the nucleus, and the factory, the ribosome, is on the outside of the nucleus. DNA cannot leave the nucleus. It's too dangerous for it to be leaving and going out directly to the ribosomes. So the DNA is stuck in the nucleus, The instructions are stuck in the nucleus, and our factory is stuck outside the nucleus. So those ribosomes, maybe they're floating around in the cell. Maybe they're attached to another organelle called the rough ER. But either way, they're outside the nucleus and the DNA is inside. So what that means for us is that we need some way. We need some sort of system to where we can get the DNA instructions out of the nucleus and to the ribosome. So we need some sort of copy of the instructions to be made so we can take it out of the nucleus, take it to the ribosome, and then have the ribosome build the protein that we need. So if DNA isn't going to do this job, what is? What's going to be our copier? Oh, right. See, sometimes I get ahead of myself. I get so excited. So the DNA code has got to be copied and then read by the ribosome, because where the ribosome is what takes the amino acids and sticks them all in the line to turn them into proteins. So this whole process, this whole system has a fancy scientific name of protein synthesis. So synthesis, if you remember from when we talked about the S phase of the cell, synthesis is just a fancy scientific word meaning building or making something. So in biology, when cells or bodies or animals build things, we call it synthesis. So in this case, we're building proteins, so we call it protein synthesis. The process of building proteins based on the information in our DNA. So we've got to copy our DNA code. All those A, T, C, Gs, we got to copy the gene and then take those instructions to the ribosome so that the ribosome can say, we need this amino acid, then this one, then this one, then this one, then this one, and we can make it into a protein. That's where our buddy RNA comes into play. So if DNA is like the original version, the master plan, RNA is like the blueprint. It's the copy of the master plan. RNA, specifically a type of RNA that we're going to talk about in a little bit, is what comes in, copies the DNA gene, and takes that copy to the ribosome. So DNA is our original master plan, the master set. RNA is the copy. It's kind of like if you're into like music at all or know people that make music, DNA is like the original recording, the master, what they recorded in the studio when they were recording the album. And then RNA is like the copy that you download from iTunes or Amazon or stream through Spotify. All right. So DNA is our original, RNA is our blueprint, our copy of the master plan. So there are a couple major differences between RNA and DNA. We've talked about RNA briefly before, but there are three major differences between RNA and DNA that I want to highlight here. So DNA, if you remember, it has a big, fancy, hard to say name of deoxyribonucleic acid. RNA is a little bit shorter name. It's just ribonucleic acid. So the R stands for ribo in ribonucleic. And I mentioned this on Tuesday when I talked about DNA and RNA. Remember, they each have that pentose sugar, that 5-sided sugar that makes up the phosphate sugar base of the nucleotide. DNA has a sugar that looks like this, where one of its carbons is attached to a hydrogen on one side and a hydrogen on one side. RNA has the exact same setup, except its carbon is attached to a hydrogen on one side and an oxygen hydrogen on the other side. So this carbon is attached to an oxygen. This one is not. So this is sort of like the original form of the sugar called ribose sugar. This one is missing an oxygen, so they call it deoxyribose. So their first major difference is in this sugar. The reason why they have the different names. DNA has deoxyribose sugar because it's missing that oxygen down here. That's where the deoxy comes into play. RNA has that oxygen, so it just has regular ribose sugar. So that's the first main difference between RNA and DNA. They have different sugars. One is ribose, one is deoxyribose. Second, and probably biggest / most important difference between DNA and RNA is that RNA does not have the base thymine. There are no T's in RNA. Instead, it has a modified version called uracil, which we represent with a U. So DNA is made out of ACGT. RNA is made out of ACG and U. So this is going to be really, really important when we talk about making the RNA strands and matching it with the DNA. Whenever you're making RNA, whenever you're making a string of RNA, there are no T's in RNAs. Instead, where a T would go, we swap it out with a U. So that's probably the most important difference, or the one that's going to come up the most when we're going through these notes and when you're doing your homework. So RNA does not have any T's, no thymines. It has uracil instead. It has a U instead of a T. Finally, the probably most obvious difference, if you're ever looking at DNA and RNA, is that DNA is double-stranded, whereas RNA is single-stranded. RNA just has one strand that kind of goes around, whereas DNA has those double strands that wrap around each other to make that helix. So RNA has one side, but not the other side. So here is a nice little picture that I think kind of highlights this. Again, we have our DNA on one side and our RNA on the other side. We've got our double-stranded, so we have one strand that goes like this, and we have one strand that goes like this. And DNA, RNA, just got our one little single strand. It's not really highlighted on here, but remember that DNA has the deoxyribose sugar, RNA has that ribose sugar, making up the background. And then most importantly, DNA has G, A, C, and T. So in this case, A and T, yellow and red, are always together here and here and here and here. And G, C, green and blue are always together here and here and here and here. On our RNA, instead of T, we swap it out for U. So we have some G's, we have some C's. No, sorry, C's are blue. We have some A's in yellow, and we have some U's. In this case, they are in orange. So DNA has T, RNA has U. So different structures to slightly different structures. T has like this little guy over here. whereas a U is basically the same, but without that little guy. You don't really need to know about structure. The main thing to remember is that there are no T's in RNA. They get switched out for U's. So we've got a different sugar, a different number of strands, and a different base between RNA and DNA. Those are our three major differences between the two types of nucleic acid. So again, on DNA, A's and T and A will always match up with each other. And then C and G and C always match up. And then when we're building an RNA, we'll match G's with C's, we'll match C's with G's. We'll put in A's can go in RNA, but we cannot have T's in RNA. We have to have a U instead. So when you're making an RNA molecule, if you're about to put down a T, stop and instead put down a U. All right, any questions about DNA versus RNA before we keep going?
No.
All right. So RNA actually comes in a couple different flavors, if you will. One of which we're going to talk about a lot, one of which we're going to talk about a little bit, and one of which we're not really going to talk about at all. So there's ribosomal RNA, which is represented with rRNA, which helps build our ribosomes. This one you don't really have to worry about. We're not really going to talk about it very much. Just know that it's a third type of RNA. There's tRNA, which has this kind of funky three-leaf clover shape, which is really important for carrying the amino acids around. So we'll talk about those a lot more on Thursday. But the big one that we're going to talk about a lot on today and Thursday is messenger RNA. mRNA is how it's written down. And this is what copies the DNA code and carries that info to the ribosomes. So the basic thing that we've been talking about this whole time, needing something to come in, copy our DNA gene, and then carry that copy out to the ribosome, that is the job of messenger RNA, mRNA. So mRNA is extremely important. It carries the message from the DNA out of the nucleus to the ribosome. And so a strand of mRNA contains the instructions for making a specific protein. So again, we have our gene on our DNA that has our specific protein instructions. The mRNA comes in, copies that gene, and then takes that copy to the ribosome. Once it gets to the ribosome, it'll read the code and turn it into a specific protein. And exactly how that works, we will get into on Thursday. So today, the main thing we're going to focus on is copying the DNA message. So the way that mRNA is set up, or the way that it copies and carries the message, is in a sequence of 3 bases called a codon. So every set of 3 bases on the mRNA is something called a codon. And we'll talk about what that does a little bit later on. So most mRNAs start with the bases AUG. Remember, we have U because there are not any T's in RNA, only U's. AUG is normally thought of as the start codon. It's what starts the mRNAs in most cases. And then in order to tell the ribosome to stop reading the RNA, stop making the protein, there are certain codons like UAA or UAG or UGA called stop codons that basically tell the ribosome, stop here, we're all done, you've made the protein, you're good to go. So we'll talk exactly about how mRNA works in a second, but it's made in the nucleus. So it's able to be built in the nucleus, and then it takes that DNA message, leaves through these like little holes in the nucleus called pores. Just like you have pores in your skin, the nucleus and your cells have pores in order to let these mRNAs out. And again, they're made up of A, G, C, and U. So there's no T's in mRNAs, there is U instead. So these messenger RNAs, these mRNAs, these are what are copying the DNA code and taking that information to the ribosome. So the way that mRNAs work looks something kind of like this. It's very similar to DNA replication, except with fewer steps. Basically, we have this kind of all-in-one protein right here called RNA polymerase. Again, this is another protein, another enzyme that's helping out. And it basically goes along the DNA, opens it up like helicase, and then matches the DNA strand that it's reading to mRNA bases. And so just like how in DNA replication, that DNA polymerase goes along and matches up the DNA with its copy, same thing goes here, only instead of matching up DNA bases, we're matching up mRNA bases. So it'll come along and it'll start building this mRNA strand. So like at the sequence we're at right now, it says, okay, we have a T on the DNA, and so we need to match that with an A on the mRNA. Move on to the next one, we have an A on the DNA, which means we match up with a T, except we don't, because this is RNA. So we don't have any T's, so instead we put a U. So an A on the DNA means that we'll put a U on our mRNA. We have a C in the DNA, which matches up with the G on the mRNA. G on the DNA matches up with C on the mRNA. So then we have another A, so we bring in a U, not a T. We have a G, so we match it with a C. We have an A, which matches with a U. A matches with U. We have a T, which is then going to be matched with an A, and so on and so forth. So this protein, this mRNA polymerase, will travel down a gene, and for each DNA base, it'll match it with the complementary mRNA base. So we end up with a strand of mRNA that has been built from this gene, built from the specific sequence of DNA. So we're going through, we're traveling through the DNA, we're opening it up, and I actually have a really good video that I'll show you guys before we end the class today that shows a lot of this stuff kind of in motion as it's going. So we go through, we copy over our DNA and we make this nice, big, long mRNA strand. Again, we have to do this because our gene is right here inside our DNA, but the ribosome, which is going to build the protein, is outside the nucleus, outside the reach of the DNA. So instead, we have to use this protein, this polymerase right here, and build out this mRNA strand so that this guy is going to be responsible for taking the DNA instructions, making a copy of them, and taking that copy to the ribosome. So that's what's going on in this first part of this process here, this first part of DNA synthesis. We're copying our DNA strand onto a piece of mRNA. So like I mentioned earlier, each set of 3 bases in mRNA is called a codon. So I mentioned that there's certain codons that are like our start codons. So like AUG is a start codon. And each codon, each set of 3 bases, 3 letters, matches up with a specific amino acid. So like, for example, AUG, the start codon, is the code for the amino acid methionine, represented by this kind of yellow bubble here. GUC, this codon, valine, matches up with the valine amino acid. AGU is the code, so that's where the word codon comes from. AGU is the code for serine, and CCA is the code for 4th amino acid. In this case, it's proline. And then the stop codons don't code for an amino acid. They just tell the ribosome, stop, don't read any more of this strand, of this code. So tomorrow, on Thursday, we'll really get into how exactly it is that the ribosome matches up these amino acids with these bases. All right, so does anybody have any questions so far about how we take this DNA strand and turn it into mRNA? All right, so before we go any further, I wanted to show you this part of this video, which I think, yes, I've also posted in the Google Classroom already. Hold on, let me actually do this. There we go. So there's not really going to be any sound. I mean, there is a nice British lady talking. You guys probably won't be able to hear it, but that's okay because I'll kind of show you the main stuff that's going on. So here we have cells. And we have a whole bunch of different structures in our cell, but the important one we're talking about today is the nucleus. So our nucleus, again, this is where our DNA is hanging out, nice and safe and protected here in the middle of the cell, right? If stuff happens to the nucleus, if that genetic code gets broken or mixed up at all, that could be really bad for the cell and for any future cells. So we're keeping that DNA nice and safe inside the nucleus. But then outside the nucleus, around inside the cell out here, they're not really shown in this zoomed out view, but we have the ribosomes. So there might be some ribosomes around here, this area. There might be some ribosomes kind of floating out in the cell over here. But that's why we have to copy that DNA information into something that can move out of the nucleus. So if we zoom way in, the DNA is usually packaged up really tight. And these things called chromosomes or chromatin, it's curled around these things called histones. Don't really worry about that. But here is our nice big DNA strand. And this certain section of it, from here to way, if you go down the strand to about here-ish, that is our gene. So that's the sequence of DNA that we need to copy and take to get it turned into a protein. So this gene right here has the code for one specific protein. Where's my mouse? There's my mouse. So our buddy RNA polymerase is going to come along, read the different parts of this gene, and as it goes through and opens and reads the DNA, it's going to match the DNA strand with a matching RNA strand. So our original base strand is DNA, but the new strand that we're going to make is RNA. It's going to have the different sugar. It's going to have a U instead of the T. And it's just going to be one single strand, not a double strand. So we're going along. We're matching up bases as we go. And again, we're still going with all of our normal base pairing rules. So wherever there's a G on our DNA, we're going to match it up with a C on the RNA. Wherever there's a C on the RNA, we're going to match it up with a G on, or excuse me, wherever there's a C on the DNA, it's going to put down a G on the RNA. Wherever there's a T on the DNA, we're going to put down an A on the RNA because A's and T's still go together. And then the only difference is that wherever we have an A on the DNA, so there's one right here and then one further down here, we have a U on the RNA. So no T's, this is an mRNA strand right here. No T's in mRNA. Instead, we do it down a U. So you can see we have a U that's coming in right here that's going to match up with our A. So basically, once the mRNA gets matched up and we get a couple of them linked together, the mRNA starts to kind of peel off. So you'll see at the edge here, it doesn't stay connected for the whole time that it's getting built. Once it gets matched up and once a couple of them are together, we can start kind of peeling it off the DNA to get it ready to transport out of the nucleus. So we're going along, we're matching up our bases. C goes with G, A means a U, C, G, A, U. G means C, T means A means U, so on and so forth. So we're matching up all of our bases. And then you can see on the edge over here that once they get matched up and a couple get bonded together, they start to kind of peel off at the end here. So it goes through, reads the entire gene, and makes a nice long mRNA, messenger RNA copy. So now this now has a copy of the original gene that was in the DNA. And this copy is what is going to leave from the nucleus and go to the ribosome to get read and turned into a protein. So don't worry about this next part. There's some other stuff that goes on with the mRNA once it gets built. There's like certain sections that get taken out and other ones that get put together. Don't worry so much about that. The main thing is that once the mRNA is all done, it goes out and leaves from the nucleus. So we have our nucleus, our DNA is staying in here. It's not going anywhere. The DNA is staying nice and safe in the nucleus down here, but the RNA can leave. The RNA is going to go out into the big wide world of the cell. And eventually that mRNA strand is going to link up with a ribosome. And so now we can finally start getting to the point where we're turning that mRNA into a protein. And that's kind of what we're really going to get into on Thursday, is how exactly this ribosome reads the mRNA, reads the different codons, those set of 3 bases, and turns it into a protein, something that's going to go out and do important work for the cell. So that's the first part of this process, that kind of how and why we need to get that DNA gene, that instruction set, copied and out of the nucleus. It's all thanks to that mRNA copying that gene and taking those instructions out to our ribosome out here. Let's go back to here. So let me move forward a little bit. There we go. So this is a picture that kind of shows the whole process. Again, we'll get into this part over here tomorrow. But this first sequence, this first series of steps is called transcription. So sometimes, and I think on the very first slide, like the cover slide on here, you saw it as well. It said from DNA to RNA to protein, and then at the bottom, in like small letters, it said transcription and translation. Transcription is that first part of the process that we talked about here today, where we're taking the DNA, we're making an mRNA copy of that gene, and then we're taking our RNA out of the nucleus into the cell. So reading the DNA and copying it into RNA is called transcription. All right. Any transcription questions from the notes so far today? Anything about this process that you want me to go back or that you're still a little confused about?
No.
Awesome. Thanks, Naya. Anybody else? Anything about transcription that you want me to go back and talk about a little bit? All right, Samyan feels pretty good about it as well. Awesome, awesome. So for the transcription related homework, I have a couple things for you guys to work on today and tomorrow. So for today, it's a fairly simple kind of question assignment. Basically, I just want you to list and describe the 3 major differences between DNA and RNA. So I talked about this quite a bit today, so hopefully you can either go back and remember or find the slide or find the part in the video where I go over it. But there are three major ways that DNA and RNA are different from each other. So for today's assignment, all I want you to do is write down what are those three major differences. Then if we go check out the assignment that is going up tomorrow. So for your daily assignment on Wednesday, tomorrow, it's very similar to one that you have had already. It's our transcription worksheet. So again, taking a DNA gene and turning it into RNA. That's what transcription is about. So we have a sequence of DNA right here. And before, you had to come up with the matching DNA sequence. This time, I want you to come up with the matching mRNA sequence. So if this was the copy of DNA that our RNA polymerase was reading, what would the mRNA strand look like? What would it look like if we used this as our template and then matched up an mRNA strand to it in order to take that out of the nucleus and turn it into a protein? So again, similar to what you've seen before, only this time, instead of making DNA, you're making RNA. So make sure to remember some of those key differences, especially about the T versus the U when you're making your RNA. So that'll be going up early tomorrow morning for your Wednesday daily assignment for your attendance check. All right, does anybody have any questions before we wrap up about the DNA, the RNA, the transcription and copying them over, or the homework for today or tomorrow?
Homework for tomorrow.
What was that?
I couldn't see what you did for the homework for tomorrow because it was like blurry.
Oh.
I can see it now.
Okay, good. So basically, it's just really similar to that earlier homework where you made a copy of DNA, only now you're turning this into a strand of mRNA. So you're saying, okay, if we had RNA polymerase come in here and make a nice mRNA strand, what would it look like if this was our DNA template?
I get you now.
Awesome, awesome. All right, anybody else? Any other questions, comments, concerns for biology today? All right, awesome. One thing that I wanted to mention here before we wrap up for today is more of a kind of a general announcement type thing rather than something specific to this class. But myself and some of the other teachers have noticed a little bit of a drop off in attendance on the asynchronous days. So like for this class, your Monday and Wednesdays. For the most part, everybody in here has been doing a really good job of keeping up with that so far. But just a reminder, please do be checking in and logging on to your Google Classroom on Mondays and Wednesdays as well. and doing those daily assignments because it's really easy to forget a couple of those and let them slip by. And then all of a sudden you have too many absences at the end of the term. So nobody wants that. We all want you to pass and graduate with flying colors from biology B class. And so just make sure, again, a lot of you guys are doing this really well already. So just keep up the good work. But for this class and all your classes, just a reminder to make sure you're logging on to those off days, those Mondays and Wednesdays for this class, and making sure you get your attendance counted for those days.